To be able to see small specimens which we cannot perceive with the naked eye, we use a magnifying glass. A magnification of up to 15 times is possible with it. First, the specimen to be looked at is captured from a particular visual angle. The magnifying glass, a convex lens, positioned between the eye and the object, reflects the light rays from the specimen in such a way that a magnified upright virtual image is created for the eye. The specimen is projected outside and the visual angle thus enlarged. Is the magnifying glass not strong enough for a closer look, we need an optical microscope. Just as with the magnifying glass, the light rays reflected from the specimen below fall onto a convex lens. This produces a magnified, reverse, real image on the second convex lens, the ocular. The latter works like a magnifying glass and now creates a new, magnified image, which is projected outside again by the eye. This, too, leads to a respectively enlarged visual angle. To obtain a better depth of field, a drop of immersion fluid is put onto the cover glass. The light rays coming from the specimen are completely reflected by the air and do not reach the lens. Is the air between specimen and lens replaced by an immersion oil? The refractive index is altered and more light rays reach the lens, thus increasing the image quality. To obtain magnifications of 1,000th millimeter, scientists use a scanning electron microscope. A cathode produces an electron beam which is accelerated by high voltage towards the anode. Beams are concentrated in the magnetic lenses and focused on the specimen. The electron beam arriving there automatically scans the specimen in a raster fashion. Secondary electrons are captured by specialized detectors and used for image creation on a monitor. The electron beam hitting the specimen activates a pear-shaped area of it. Through interaction of the arriving electrons with the atoms of the sample, secondary radiation is produced, which is used both for the creation of images and for research into the specimen. The diverted electrons are collected for the sample to be recharged. The atomic force microscope, also called scanning force microscope, is able to provide three-dimensional pictures in the nanometer range, that is at millionths of millimeters. With the help of this technology, we can render the red blood cells in this drop of human blood three-dimensionally visible, like in our example. The core element of this microscope, an atom-sized point of a needle fastened to a pliable leaf spring called cantilever, is positioned above the object. At the right distance, the point scans the surface of the blood cells. Via the computer, a three-dimensional image is created, which makes every detail of the blood cell visible to us. We are taken to an entirely new world of microcosm.